So our next speaker is Dr. Abigail Renegar. She's a research scientist at Nova Southeastern University's College. Thank you. She's been at NSU for over 10 years and presently manages the Marine Toxicology Laboratory, Coral Histology Laboratory, and land-based coral nursery at NSU's Center of Excellence for Coral Reef Ecosystems Research. Her primary research goals focus on experimental evaluation of the impacts of land-based sources of pollution and climate change on scleractinia, coral biology, and ecology. Her present research includes multi-year projects examining hydrocarbon toxicity to scleractinian corals and mesopelagic crustaceans. So, without further ado.
Thank you. Thank you, Monica. So what I'd like to do today is try and share with you a little bit of some insight into my experience with hydrocarbon research, into how one might go about developing and designing a collaborative study that attempts to bridge that gap between oil spill science and good science and relevance to oil spill response. So for the past several years, I have been co-PI on the NSU CCA Coral Hydrocarbon Toxicity Study, which really grew out of a need to address data gaps relating to the sensitivity of scleractinian corals to hydrocarbons. So what this study really represents is a collaboration between academia, between government, between industry and responders. It was really designed from the beginning with the end in mind in order to support the decision-making process should a spill potentially impact a coral reef environment. So the complex nature and solubility of oil and petroleum hydrocarbons really presents some challenges for toxicity testing, and working with corals in any capacity can be difficult as they can be quite tricky to keep in culture. So as researchers, it was really our job to design exposures which considered both the characteristics and the requirements of corals and use protocols and metrics that were specifically designed for scleractinian corals, which considered both acute and subacute effects at different levels of resolution. All the while working to ensure that the data that we produced was compatible with current and emerging fate and effects models that are used and applied to oil spill response. So the genesis of this project, as I think is true for most projects, was the recognition of a knowledge gap. For us, it was regarding the sensitivity of corals to hydrocarbon exposure, which could impact the ability to assess risk in the event of an oil spill impacting a reef environment. So I think as academics, the approach that we're most familiar with is the RFP approach. So some agency puts out a request for proposals, and either a researcher or a group of researchers gets together and comes up with some interesting work to do to sort of address the goals of that RFP. But this was a little bit different. So Paul Schuler, representing Clean Caribbean and Americas, contacted, member of the response community, of course, contacted Dr. Richard Dodge at NSU, who is my dean, about developing and funding a research program to address this data gap. Dr. Dodge, in turn, contacted Tony Knapp at Texas A&M. So from the start, we really recognized the benefits of directly incorporating some collaborative input, both from the response community, from industry, and from government, by means of a small oversight committee and a larger group of collaborators. So our oversight committee included members of the response community, namely Paul Schuler, also industry, so we had representatives from ExxonMobil, from Shell and from Chevron, as well as government, Brad Bengio from NOAA, and Cedra in France. So we sort of got together, discussed some ideas. We had a number of discussions, a number of meetings, and we were given some initial tasks. So the first was to develop, or begin really, a literature review, and then start to develop some initial ideas for some experiments or a series of experiments. So one of our first tasks was a review of existing oil spill research on corals which ultimately included about 44 years worth of studies. So these previous studies can generally sort of be divided into three categories. The first of these is spill incidents, which have resulted in acute or chronic exposure of corals to petroleum in some way. Now, every spill event presents some unique challenges due to the large variation in environmental factors, biological, chemical, physical conditions associated with each spill, as well, of course, as variability in the oil itself. So in situ field and ex situ laboratory studies are designed to allow researchers the ability to control the exposures and any compounding stressors that there might be, and limit differences between exposure scenarios as a means of comparing toxicity between different corals and different oils. So for spill incidents, understanding impacts is really complicated by a frequent lack of baseline data, as well as variability in environmental conditions, as I said, and that really leads to a wide range of variation in observed effects. In situ field studies, Paul talked a little bit about the tropics study earlier, can include actual exposure concentrations, as well as baseline coral community data, but for obvious reasons are quite limited in number. So for ex situ laboratory studies, which by far represented the largest chunk of the studies that we reviewed, we find a tremendous amount of variability, not only in study design, by that I mean species tested, the toxicant utilized, the rate of exposure, and any other bioassay condition that you can really think of. What all of this variability results in is reported exposure effects that range from no effect at all to complete mortality, with a variety of sublethal effects reported in some studies, but not in others. And of course, one of the most important issues is that characterization of the exposure media was quite frequently lacking in most of these studies. In fact, only about 20% of the studies in this review used any sort of measured concentration whatsoever, and that can really cause problems in terms of how your endpoints are interpreted. So based on our literature review and feedback from our research partners, we began the process of designing and refining our experimental design and protocols. So I want to stress that this was very much not a linear process. This sort of feedback loop went round and round several times. I can tell you that sort of what the core academic group, which is us and Texas A&M, started out thinking that we might plan on doing was not what we ended up doing at all, and it very much for the betterment of the entire process. So several repetitions of this, and we finally had an experimental plan that was going to work. So the result of all of that was a phased research program. The first phase included completing the literature review, which we had done, and developing test protocols and an experimental design that reflected the considerations and some of the issues that we'd identified in previous research. The second phase focused on assessing toxicity of individual hydrocarbons to model toxicity of whole oils, and this was something of a novel approach, at least for us. And in the third phase, we would then take our predictions that we had generated with our single hydrocarbon studies and attempt to refine the accuracy of the model by using whole oils to do our toxicity studies. So really what we were looking to do was use the single hydrocarbon information to predict the toxicity of a particular oil based on the assumption that corals would conform to the predictions of that model. But then we, of course, had to test that by using oil and subsequent to that, CWAF as well, so chemically dispersed oil. So why did we decide on this? Why did we decide that the types of experiments in phase 2 and 3, how might data of this type be used in oil spill response? So toxicity data is very often used to generate SSDs, or species sensitivity distributions. So these are a cumulative distribution of toxicity endpoints, which allow the comparison of relative sensitivity of different species to the same chemical. So these represent some scientifically defensible benchmarks, typically at the same exposure duration, as verification of consensus values for levels of concern. And these are applied in evaluation of risk to specific resources in NEBA and SEMA activities. So modeling toxicity is a significant part of toxicity testing today. And based on the assumption of additive toxicity, it's possible to predict the toxicity of any oil based on the toxicity of its constituent hydrocarbons. So that was sort of the context for the applicability to oil spill response of the data that we would be generating. So this is what we came up with. So our exposure design for the individual hydrocarbons testing used passive dosing of our single hydrocarbons. We used 1-methylnaphthalene, phenanthrene, and toluene via PDMS O-rings, silicone O-rings. This type of passive dosing has some implications in terms of maintaining constant concentrations over time, which can be difficult to do with volatile compounds, and that has some real implications for modeling. Very important to us, each of our experimental exposures consisted of a pre-exposure condition in which our organisms were acclimated to the laboratory and we were able to collect baseline data. We then had our 48-hour constant exposure period. And then after the exposure, we moved our organisms back to clean water to assess any potential for recovery or delayed effects after the exposure. And of course, we have done quite a lot of very expensive characterization of our exposure media in terms of hydrocarbons. So how did we assess the coral? So there's really no blueprint for this in terms of a toxicity testing protocol for corals. What we ended up with was sort of a modification of some previous work that we've done looking at sedimentation effects on corals. So what we do is we evaluate the physical condition of the corals using a semi-quantitative 4-level scoring system, with 0 being within sort of normal limits, what we would expect for that species, and 3 being quite severely affected. So we consider changes in coloration, in polyp extension and retraction, tissue swelling, tissue thinning, attenuation, mucus production, in addition to the normal endpoints of mortality and partial mortality. Now, tissue thinning and attenuation is something we have actually added based on subsequent experimentation to our initial protocol, based on our observations of different species of coral as we went along with these experiments. We also measure growth rate and photosynthetic efficiency throughout and assess cellular changes histologically. So subacute endpoints like these are extremely important because they relate ecological effects to durations and concentrations that may be more realistic in real world scenarios, but that might have significant impact. Oops. So the next step in that process was to do our experiments. After each experiment, we generated an experiment report that was shared with our collaborators to generate feedback from them that we could apply to upcoming experimentation so that we could continually improve our methods and make sure that we ironed out any difficulties as we went along. So I just want to share with you a few observations of the type of physical response that we typically see during the exposures. And this is all 6 species that we have worked with so far in the laboratory. So a combination of branching and mounding or massive species. And we found that these guys really exhibit a range of response to pH exposures with a sometimes significant degree of variability between species. So one of the most obvious responses, the pointer here, is polyp retraction. This is what corals do. You disturb them in the field or the laboratory. The first thing they do is pull their polyps in. Second, response, and it's difficult to see here, but mucus production. So corals have significant capability to produce and release mucus. They have large mucocytes in their epidermis and elsewhere. And this is something that they do all the time. Corals there's some degree of variability between species, but they're always releasing this lipid-rich mucus. So they can produce so much mucus that you actually see, you can kind of see it in that one, pooling around the base of this coral fragment. Very interesting response, something that we hadn't necessarily expected to see, was tissue swelling. So I've got two examples of this, both in a massive coral, this is Siderastrea siderea, and in a branching coral, this is Parietes divaricata. So we tend to see this at lower concentrations. So this top row here is sort of what a normal coral looks like for that particular species. And you can see here in Siderastrea, this tissue is all swollen up. And in the branching coral, again, this is divaricata, this is what it would normally look like. And we saw this incredible degree of tissue swelling and, more importantly, polyp distension in the species. And this is really important as a sublethal effect because, as I said, one of the first responses of a coral to any sort of disturbance is to pull its polyps in. A coral that has this level of distension and swelling is not capable of doing that. So whereas you can disturb a normal coral, they will immediately retract their polyps. These guys can't do that. So it's either a delayed response or they don't do it at all. Which means that the tissue is more subject to damage, more subject to predation, that sort of thing. Then, of course, we do see bleaching. This is Stephanocenia intercepta. Believe it or not, that coral's alive. It just doesn't have any of its endosymbionts anymore, really. And then, of course, the endpoint for all of this is mortality. This is a normal Parietes asteroides, and this is a fragment that's experiencing that white area is some partial mortality. Now, here's where corals are a bit different from other organisms like shrimp or anything like that, is that you can, it's easy to tell when a shrimp is no longer alive, right? It sort of falls to the bottom and stops moving. Corals don't do that. You will also occasionally see, the whole piece won't have mortality, but you'll see partial mortality. So only part of the coral will die, and if you move that back into clean water, it can regenerate. So we saw that quite often. Even if we would lose part of the fragment, it would grow back once it was transferred back to clean water. So it's just some of the examples of what we typically see. So we've done our experiments. The next step is really to share that data via conferences and publications, sort of what I'm doing right now, thus making our data available for inclusion in risk assessment and thereby overall contributing to an improvement in effective decision-making should a spill impact reefs. So the success of this project, and more importantly, the success of this process, led to two additional projects, both of which were funded by the Gulf of Mexico Research Initiative. So the first of these was the Detox Project. For this project, the main objective was really to provide some new hydrocarbon exposure thresholds for mesopelagic crustacean species. But we used a very similar approach to evaluate toxicity, initially, again, with the single hydrocarbons, and then with oil and dispersed oil for both mesopelagic species, and then afterwards also with shallow water proxy species. So for this particular project, we also focused a great deal on development of improved methodologies for oil exposures, specifically passive dosing of oil, working with Aaron Redman and Tom Parkerton at ExxonMobil. So subsequent to the Detox project, the Coral Tox project was also funded. So this project really grew directly out of the original CCA project and was designed to expand on our existing work with corals by providing new data for additional species in order to provide sort of a more complete picture of where corals fit on the scale of sensitivity to hydrocarbons compared to other species. Research plan was very much based on previous work. Again, the goal was to continually expand and refine the testing protocols that we had previously developed. So back to our original goal of connecting science to response. So before I conclude, I kind of wanted to share with you what this type of data looks like and what you can subsequently do with it. So it's a bit odd standing up here to see sort of the past couple of years of your life summarized in one table on one slide. There's a bit more to it than that, I'll say that, but this is quite a lot of man hours involved in generating this data. So these types of experiments generate both effect and lethality data. So here are all the species that we've worked with. This is the effect concentration data in terms of concentration of the chemical, and here's the lethality data over here. So that endpoint data can in turn be used to calculate what's known as a critical target lipid body burden, which is the organismal tissue lipid concentration of a specific hydrocarbon that results in morbidity and eventual mortality for the organism, okay? So I've highlighted both the EC and the LC calculated body burdens for our original test species, which is Divericata. So there are three chemicals at play here. This is 1-methylnaphthalene, that's phenanthrene, and that's toluene. And we can see that the EC CTLBB for each of these different chemicals is pretty similar between them for the same sort of endpoint. And the CTLBB that generated using the lethality endpoint is also the same between 1-methylnaphthalene and toluene. So this enables you to compare different hydrocarbons between species and within the same species. So the endpoint data, i.e. the EC50 or the LC50, can be used to compare species sensitivity distributions, as I mentioned previously, such as this one, which has been generated by NOAA's CAFE database. You can also use the CTLBB data to compare the relative sensitivity using similar data for other species that has been determined for multiple hydrocarbons. So for the corals over here, we can see that with the exception of Acropora cervicornis, the staghorn coral, which is that one right there, the corals appear to be relatively resilient to hydrocarbon exposure, although there is a range of species-specific responses with greater impacts generally observed in branching species versus massive species. Now, Acropora cervicornis is the most sensitive thus far. Of course, all corals are protected. Atlantic corals are protected in the United States. But cervicornis is a bit special because it is listed as a threatened species under the Endangered Species Act, and that has some implications in terms of how you consider that species in NEMA and SEMA activities. Okay. So before I conclude, I just wanted to share some thoughts on the lessons that I've learned from the development and conduct of these projects. So literature reviews are key. Identify data gaps. What do we need to know? What do we want to know? It helps you to identify best practices, how to conduct the study and provide the information that's needed. Also, very importantly, what not to do. If you're working with a new species, as we were with coral, doing range finding tests, so limited concentrations to really understand the range of response of your organisms are very important. Also designing with the end in mind. A bit like statistics, right? You need to sort of know what you're going to do in order to conduct the experiment properly. Identify upfront the practical application of your results. And of course, collaborate with research partners and end users. So recognizing data gaps and being willing and able to collaborate with partners outside academia can lead to new opportunities to develop more meaningful research. It's also very important to have the ability to adapt your research to reflect new information that you gain along the way, because of course the research process can answer questions with more questions. And how to do this is frequently sort of a judgment call on the part of the researcher, but the applicability of that can be evaluated with input from your collaborators. But what I'd really like to leave you with, and to take away from this, is that overall the collaborative process really serves to promote a greater understanding of how to improve oil spill response as a whole by making scientific studies more applicable and directly relevant to response. I know for my part, this work has given me the opportunity to participate in some NEBA activities, and that for me was unbelievably important in understanding how my data was utilized and how I could best tailor that data to make it as useful as possible. Okay. So thank you all very much. I've got to thank Clean Caribbean Americas and GOMRI for their funding, and all of my collaborators and my graduate students working with them. Thank you.